Albumin. Human serum albumin, 585 amino acids, 66.4 kD, is an extremely soluble protein. Its normal level in plasma does not exceed 5 grams per 100 ml, but solutions containing 50 grams per 100 ml can be prepared in vitro. Albumin adopts a heart-shaped tertiary structure with 2/3 of its residues in 28 alpha helical regions. It has 17 disulfide bonds, an unusually large number. Unlike many extracellular proteins, albumin is not a glycoprotein. Albumin serves 3 principal functions. First, albumin stabilizes the physical environment of the blood in 2 ways. By providing 80% of the osmotic pressure of plasma, albumin contributes significantly to maintaining the osmotic balance between blood and cells, and as a major macromolecular anion in plasma, albumin supplies most of the acid-base buffering action of plasma proteins. Second, albumin is a major transport vehicle for hydrophobic substances, carrying fatty acids from their sites of storage in adipose tissue to their sites of oxidation in liver and muscle. Albumin also transports bilirubin and heme from the spleen to the liver, steroids from the adrenal glands to other tissues, and thyroxine from the thyroid gland to its target tissues. Third, albumin serves a protective function by binding toxic metabolic byproducts, waste, and ingested substances including bilirubin, aflatoxin, therapeutic drugs, and xenobiotics such as benzene. The ligand binding capacity of albumin is quite broad. Because albumin binds smaller molecules of many types and thus carries almost any cargo, it has been called the tramp steamer of the blood. Hydrophobic organic anions of medium size, such as long-chain fatty acids, eicosanoids, and bile acids, and steroid hormones are most strongly bound. For many of these compounds, albumin acts as a storage depot. Such substances, when bound to albumin, become available in amounts in excess of their solubility in plasma. Potential toxins are also bound and sequestered for transport to disposal sites. Albumin has 11 distinct fatty acid binding sites, and circulating albumin molecules typically have 1 to 4 long-chain fatty acids bound. Long-chain fatty acids bind in hydrophobic pockets with their carboxylates forming salt linkages with nearby lysine or arginine. By binding to albumin, long-chain fatty acids reach a concentration of about 1 millimole in plasma, despite an intrinsic solubility of only 0.1 nanomole. Although plasma contains steroid-specific binding proteins, the amounts of albumin are much higher, and thus most steroid hormone in plasma is bound by albumin. Binding is weak, so hormones are readily delivered to their targets. And that is albumin. Have a great day.